In this video, we'll look at the Antibody Developability Metrics data function in ChemCharts. The Antibody Developability Metrics data function calculates a set of metrics published in the paper 5 Computational Developability Guidelines for Therapeutic Antibody Profiling by Revvity et al. The input columns for this data function must be specifically prepared for the data function to operate correctly. A structure column containing compressed PDB files for each antibody and an associated antibody sequence column with certain annotations present are required. A typical workflow to generate these columns starts with a sequence column containing antibody protein sequences. Structures can be predicted using the antibody structure prediction data function. The data function will produce a column with predicted antibody structures as well as a sequence column with IMGT numbering and Lefranc CDR definitions. Using these columns, relative accessible surface area, or RASA, can then be computed and annotations added to the sequence. The resulting sequence column with IMGT numbering, Lefranc CDR definitions, and RASA annotations along with the associated antibody structure column together are the inputs for the developability metrics data function. This process of column preparation can be done as I've described here, or it can be streamlined. The antibody dashboard data function starts with just antibody protein sequences, predicts their structures, numbers the sequences, computes RASA, calculates developability metrics, and annotates sequence liabilities. Check out the video for the antibody dashboard to see it in action. For this example, I have prepared a table with predicted antibody structures, numbered sequences, and computed RASA for 242 commercial antibodies included in Supplemental Table 2 from the Revvity paper. In the data function settings dialog, select the data table, the fully annotated sequence column, and the associated structure column. When the data function is complete, 13 new columns are added to the table. 7 columns consisting of the number of residues in each CDR and the total number of CDR residues. 4 columns of computed metrics including patches of surface hydrophobicity, patches of positive charge, patches of negative charge, and structural charge symmetry. The metrics are described in the Revvity paper, and the algorithms were re-implemented in ChemCharts, maintaining a high correlation with values in the publication. Based on the adjusted cutoff values for each metric, yellow and red alert counts are also calculated. The table of computed developability metrics can then be used to evaluate and prioritize antibody sequences. Thank you for watching, and check out our other ChemCharts data function tutorials.